Okay, we're going to start with binary fission. And with binary fission, I like to think B for bacteria. So this process takes place in bacteria, which is a prokaryotic cell. So it's used in prokaryotes for reproduction. It's usually a pretty fast process because these are very simple cells. So the process can occur in some of them in about 15 minutes. Others it takes longer, but it's still pretty quick comparing it to eukaryotic cell division. Now this one makes two identical prokaryotic cells. So that makes this an asexual process. Whenever we're making identical cells, it's going to be asexual. Here's what the process looks like. So we start with one bacteria cell. We are going to copy the DNA. So the DNA is replicated. The cell membrane and wall kind of start to pinch in. This cross wall forms. And then they split, again making two identical cells. So two identical bacteria cells. Next up, we have cell cycle and mitosis. This is also an asexual process because we're going to make identical cells. This one occurs in eukaryotic cells, which means they have a nucleus, so it's a little bit more complicated and there's more steps involved here. So in multicellular eukaryotes like us or other animals or plants, it's going to be used for growth and repair. So this process makes you bigger than you were when you were little, or if you break a bone or get a cut, this is what your body will do to repair the damage. And it's used for reproduction in unicellular eukaryotes. This process is going to make two identical, what we call somatic or body cells, and they are diploid or 2N. So N is the number of chromosomes. So when you have a diploid cell, you have two copies of each chromosome. You get one copy from mom, one copy from dad. And in humans, the diploid number is 46. So you have two sets of 23 chromosomes to make 46 total chromosomes. So some examples of cells that would be made through mitosis would be skin cells or muscle cells, basically any of your body cells. So anything except egg and sperm. Now, here are the phases of the cell cycle. So we're going to start over here with interphase. And interphase has three parts. It has G1, S, and G2. And for interphase, I like to think grow, copy, and prep. So in G1, the cell is growing. In S phase, we're going to copy the DNA. So the DNA will be replicated. And G2, it's going to prepare to divide. So it's going to go through a couple checkpoints, make sure everything was copied correctly. It has everything that it needs to divide. And if it doesn't, it will go back and fix those things before it divides. Now this part is where it's actually dividing. This is mitosis, and it consists of PMAT. So prophase is first. And prophase, I think pack. So the DNA is going to condense or pack into chromosomes, and chromosomes are just kind of a way to help organize the DNA. After prophase, we have metaphase. And metaphase, I think M for middle. The chromosomes line up right in the middle of the cell, or the equator of the cell. After metaphase, we have anaphase. And I think A for away or apart. The two sides of the chromosomes will actually separate and they will be pulled to opposite ends of the cell, so they're moving away from each other. And lastly, we have telophase. And telophase, I think T for 2. So the two nuclei will start to reform. I forgot to mention in prophase over here, the nucleus will also start to dissolve. So that's what that dotted line is representing. So that's PMAT, prophase, metaphase, anaphase, telophase, which is mitosis. And then after that, we have cytokinesis. And I think C for cut. So this is actually the division of the cytoplasm, and that's when they split. So here, this is where we're going to end up with two identical cells. And again, they're somatic or body cells. So with this process, we're starting with a diploid or 2N cell. So in humans, that would be 46 chromosomes. And after this process, we end up with two cells that are both diploid or 2N. Because here we copied our DNA, so we doubled it, so when they split, each one gets a copy of the DNA. Now down here we have this wheel, which is basically showing you the exact same thing that we just talked about here, just in a wheel form. So again, interphase, we have G1, S, and G2. So G1, I think, grow. In S phase, we're going to copy the DNA. Okay, and again, this is really important because every cell needs a copy of the DNA. And then G2, we're going to prepare to divide. So grow, copy, prep, which is all part of interphase. Now, this is the longest part of the cell cycle. As you can see, it's going to take up the majority of the time here. So most of the time, cells will be in interphase. Once it starts dividing, that's mitosis. So over here, I'm just going to put divide. So grow, copy, prep, divide. And again, that's PMAT, so prophase, metaphase, anaphase, telophase. And then when they actually split, this is cytokinesis. So since it's a cycle, after cytokinesis, they'll go back to G1. Now, sometimes after cytokinesis, they'll go into this G0 phase. And this is kind of a resting phase for cells. So some cells will stay in this resting phase if they're not going to be dividing, especially like nerve cells will stay in G0 forever because they don't divide anymore. Now, when this process becomes unregulated, that is what leads to cancer, which means that they're just undergoing mitosis over and over and over and over again, which is why a tumor grows. So there's certain things that can cause it, like chemicals or UV light or radiation, which basically changes the DNA in the cell so that it doesn't have a regulated mitosis and it just continues to go around and around. All right, on the next page, we've got meiosis. And meiosis is also used in eukaryotic cells. This one, however, is a sexual process because we're not going to make identical cells. So it is used for sexual reproduction. In multicellular eukaryotes. Now, this only occurs in multicellular eukaryotes because it's going to make what we call specialized cells. And to have specialized cells, you have to be multicellular. So this one, it makes 4 genetically unique haploid, or N, meaning they have 1 copy of each chromosome, gametes. And gametes are sex cells. So in humans, those gametes would be egg and sperm. And in plants, they would be ovules and pollen. Now, we want genetic diversity in a population because it's going to lead to a better success of that population. And there's 2 important processes in meiosis that occur that help create this genetic diversity for each gamete. The first one is called crossing over. And this occurs in prophase 1 of meiosis. So meiosis is going to go through cell division 2 times. So it has PMAT1 and PMAT2, or prophase, metaphase, anaphase, telophase 1 and 2. So this process occurs in prophase 1. And what happens is we have pairs of homologous chromosomes, meaning they share the same genes. They actually switch genes with each other so that each one, when they separate, has a different combination of genes. So you can see we started with one that had the same genes on both sides, and now each gamete is going to have a different combination of genes. So this increases genetic diversity. The other important thing that happens here is what we call independent assortment. And this basically just says that genes are independently inherited from one another. So they're not going to be linked to other genes. You're going to get them independently of one another. Here's what the process of meiosis looks like. So we're going to start with a diploid cell, which again is 2N, which means you have 2 copies of each chromosome. It's going to go through PMAT twice. Okay, here's that crossing over that I showed you that's occurring in prophase 1. So the DNA is replicated. So when these split, they're still diploid. Now it's not going to be replicated again. So when they split, now each one is going to be haploid. So we end up with 4 genetically different gametes. Okay, so this would be 4 eggs or 4 sperm. And each one is haploid, or N. So each one has 23 chromosomes. And the reason that we want them haploid is so when the process of fertilization occurs, we have the correct amount of chromosomes. So here is a sperm, which is a gamete. So it's going to be haploid. And here is an egg, which is a gamete. It's going to be haploid. So again, in humans, that number is 23. So each egg and each sperm has 23 chromosomes. So when they come together to form the zygote is going to be diploid, which is 2N. And this would have 46 chromosomes. So it would have the correct amount of chromosomes. Now, once the zygote is formed, mitosis will actually grow the baby. So mitosis is going to grow the baby. So again, meiosis only makes the egg and the sperm. Once they come together to form the zygote, mitosis takes over. And last process is going to be DNA replication. And I like to have 4 colors when I talk about DNA replication. So if you want to pause the video and go grab 4 different colored pencils or markers or whatever you have. Doesn't matter what color you have. I'm going to start with green, and I'm going to follow my original strand, which is up at the top, and I'm going to follow it all the way down on each side here. And then I'm going to take my second color, which I'm going to do an orange, and I'm going to color or follow the new strand, which is just at the bottom here. And I'm going to take my third color, and I'm going to color this enzyme. And my fourth color, I'm going to color this one right here. Both of these enzymes right here. Okay, so we're going to start by labeling whatever you colored at the top. This is our original or old strand. So this is the one that we're starting with. And the first thing that happens is we have an enzyme called helicase. And what it does is it goes up the DNA and it splits the hydrogen bonds that holds the DNA together. And it's going to unzip or split apart the DNA strand. Okay, so you can see here it's breaking those hydrogen bonds, so half the DNA is going here and half of it's going here. Then we have an enzyme called DNA polymerase. And what it does is it takes free-floating nucleotides and it builds the new DNA strands. Okay, so we have 2 of them because we're building 2 strands. So on here, whatever your bottom color is, on mine it's the orange, this is your new strand. And then again, whatever color you used at the top, this is your old strand. So half of it's going here, half of it's going here. Now this process makes 2 identical DNA strands. And if you remember, this process was used in binary fission, mitosis, and meiosis. So it's a very important process because anytime we make a new cell, those cells need a copy of the DNA. So this is how we make more DNA. Now, we say that this process is semi-conservative because we are sort of conserving the DNA because half of it goes here and half of it goes here. So each strand is 1/2 old. Okay, so that's the half that went here and 1/2 that went here. And 1/2 new. So on mine, that would be the orange ones. Okay, so we're conserving half of the original strand in each one that's built. And now sometimes mistakes happen during DNA replication, and when those do, those are called a mutation. So a mutation is just a change in DNA. And mutations can be good, bad, or neutral. So we usually think of them as a bad thing, but they're not always bad.